Hello everyone. Today we are going to start the chapter genetics, and the lectures are going to be really short because I don't want you to get bored in just one lecture. So every lecture is going to be short and the explanations are going to be simple so that you get to understand every single topic easily. Okay? So here we are starting with the term genetics. So, who gave the term genetics? It was given by William Bateson in 1905. What is genetics? Genetics is the branch of biology which deals with the study of genetic variations, genes, and heredity. So, you know about genetic variations, right? What are genes? Are the unit of genetics. The portion of DNA which is going to code for mRNA, okay, which is going to express some protein, that is genes. Okay. And what is heredity? So heredity is the process of passing characters. Suppose this is a character from one generation to the next generation. So it is the transmission of traits from one generation to next generation. Okay. Now we're coming to Mendelian genetics. So who gave the Mendelian genetics? Of course, Gregor Johann Mendel during 1853 to 1863. This is a really important date. I hope you'll memorize this, okay? So he did his experiment on pea plant, which is also called Pisum sativum, the scientific name. What are the characters that Mendel selected? So the characters that Mendel selected were seed shape, which are round and wrinkled. Seed coat color, which can be white, gray, brown. This one is not very important, okay? Seed color of the seed and cotyledons. What were they? They were yellow and green. Flower color, which was white and violet. Ripe pod shape, which was inflated or wrinkled. Unripe pod shape, unripe pod color. Here, not the shape, but the color. Okay, right. So the unripe pod color will be yellow and green. Flower position will be eggshell or terminal. Okay. Length of stem, tall, which is most common, and dwarf. This is the most common character that we use to demonstrate the Mendelian genetics. For monohybrid cross, I'll tell you what is monohybrid cross later. Okay. First of all, I'm telling you something that's important. So, what is homozygous? When there is two identical alleles. What are alleles? Alleles are the character or the trait that we have. For example, tall or dwarf. So this is the symbol for tall, this is the symbol for dwarf. Okay, so these are alleles. So if we have same allele, the identical two alleles together, for example capital T and capital T, which is for tall, which codes for tall phenotype, and if it's small t, which codes for dwarf phenotype. So if we have these same allele together, identical alleles together, then it is homozygous. Okay, this is not homozygous, so I'm gonna remove that. And in contrast to that, we have heterozygous. So what's heterozygous? When we have two different alleles, one is dominant and one is recessive. I'm going to tell you what's dominant and what's recessive later, so just remember what I just said. So we have two different alleles, for instance the capital T and the capital small t, okay, which codes for the tall phenotype. I'm gonna tell you why it codes for tall phenotype, because we have here the small dwarf one too. Okay, later on I'll tell you everything, you'll understand everything soon. Now let's come back here. So the factor, the unit that inherited from one generation to the next generation, this unit is called factor by Mendel. Okay, so Mendel called this unit factor. Okay, here it is written, unit of inheritance factor. This is what Mendel called. Okay, now let's come to Mendel's law of inheritance. So Mendel gave three laws of inheritance, first of which is law of dominance, which says that the characters are controlled by discrete units that we just talked about, discrete units as factor. So factors occur in pair, just like I told you, like capital T, small t, or capital T, small t. Okay, so in this dissimilar pair of factors, one member of the pair dominates the other. For example, we have small t here, but with this small t we have capital T. So this capital T is going to dominate over this small t and express itself, which means that this small t is not going to express itself because of the presence of this capital T, which means this capital T is dominant. Okay, I hope you understood. So in a dissimilar pair, I'm talking about dissimilar pair, okay, not the similar ones. When we have the dissimilar pair, so one of the member of the pair dominates the other. So in the law of dominance is only this much, which is explained by Mendel, but we can also explain it in this simple terms. Now, some alleles are dominant while others are recessive. Okay, some alleles are dominant while others are recessive. So, an organism with at least one dominant allele will display the effect of the dominant allele. Did you understood? An organism, okay? For example, this organism has capital T and small t, so it has at least one dominant trait, which is this capital T, and one recessive allele, which is this small t. Okay, so at least one dominant allele will display the effect of the dominant allele, which means this organism, this plant, will say, because we're using tall and dwarf, so this plant which has capital T, which is the dominant allele, will express this allele, which means this plant which will become tall. This plant will express itself as tall. Okay, so this is what law of dominance says. Now let's come to law of segregation. So what law of segregation says that during gamete formation, the allele for each gene, I've written gene twice, let me correct that, each gene. Okay, so during gamete formation, the allele for each gene segregate from each other so that each gamete carries only one allele for each gene. Did you understand that? Okay, let me explain it in easier terms with example. So during gamete formation, so here is the plant. Let's take the example of plant. Okay, plant. ठीक है. So this plant is forming a gamete. So a gamete is formed by a cell which has, for instance, diploid number of chromosomes. Okay, so when this cell forms a gamete, it forms a gamete which has haploid number of chromosomes, right? Now, when you had a diploid number of chromosomes, you had two pairs of chromosome. So one of which is going to be in this gamete and one of which is going to be in this gamete, right? So uh, what law of segregation says is, during gamete formation, the allele for each gene. Now, for instance, the allele for capital T is present here and capital small t is present here. Now, the allele for each gene segregates from each other, meaning it gets separated. One comes here, the other comes here. Okay, it gets segregated or separated from each other. So, each gamete, each of these gametes, these are the two gametes, each of these gametes are carrying only one allele. For that means this gamete is having either T or small t. This gamete will have either capital T or small t, not both of them. Okay, so these alleles from the parent cell uh, are going to segregate independent. I'm not saying independently because it's going to be the next law. So it's going to segregate during gamete formation to give its allele to their its two daughter cells separately. Okay, so only one allele is going to come in the the gamete. Okay, let me explain again. So here's the parent cell which has two alleles, capital T and small t. Now it forms two daughter cell. So these two daughter cell will have only one of the two alleles. For instance, this one has capital T and this one has small t. Okay, this is what law of segregation says. Let's read it again. During gamete formation, the allele for each gene segregates from each other so that each gamete carries only one allele for each gene. Okay. Now let's come to law of independent assortment. That's all for today. I'm not ending. I'm just saying that this is all for today's. This is the last topic. Okay, which is law of independent assortment. So we just learned that these two chromosomes are going to segregate. Okay, so these chromosomes are carrying the genes capital T and small t. So these are going to segregate during gamete formation and form two gametes which will carry one of the two alleles. Now, what law of independent assortment says that during this segregation, these both of the alleles segregate independently, meaning the segregation of any of these allele will not depend on each other. Meaning this tall allele will not depend on this dwarf alleles segregation, and the dwarf allele segregation will not depend on the tall allele segregation. This is what law of independent assortment says. So that is all for today. Okay, let me tell you what tomorrow's topic is going to be. So tomorrow's topic, it's going to be monohybrid cross. Okay, thank you so much.